evening. Very good evening to all the participants and respected resource person, Dr. THIRUNAHARI UGANDHAR sir, Associate Professor, Kakatiya Government College, Autonomous, Hanmakonda, Warangal. Sir is going to explain about the RNA structure and types and functions about the ribonucleic acid. It is a most important nucleic acid in the all living organism from prokaryotic to eukaryotic organisms, and also it is involved in the protein synthesis, and also it is acted as a genetic material in different viruses. Some of the viruses like polio virus, HIV virus, and recently we are observed the pandemic situation, COVID-19, SARS-CoV-2 also contains this RNA as a genetic material, and also RNA, it is also involved most of the biochemical reactions in the genetics. So Sar is going to explain in detail. So we would like to invite Dr. Tirunahari Yugandhar sir. Please welcome sir and start your session sir. Please welcome. Sir, start చేయండి sir. Welcome sir. సార్ మీరు అన్మ్యూట్ చేసుకోండి యుగేందర్ సార్, అన్మ్యూట్ చేసుకొని మాట్లాడండి. యుగేందర్ సార్ స్టార్ట్ చేయండి. సార్ స్టార్ట్ చేయండి.
అదేనా తక్కువ సార్, ఒకలే ఉన్నారా? ఓన్లీ వన్ స్టూడెంట్ ఆర్ దే?
సార్ జాయిన్ అవుతున్నారు, మీరు స్టార్ట్ చేయండి. నెక్స్ట్ ఇంకో క్లాస్ ఉంది.
So versus also DNA, both are called it can be single stranded and double stranded. Also two are different editions. So this is a
సార్ మీరు మ్యూట్ లో ఉన్నారు, అన్మ్యూట్ చేసుకోండి. సార్ అన్మ్యూట్ చేసుకోండి సార్. యుగేందర్ సార్ అన్మ్యూట్ చేసుకోండి. సార్ అన్మ్యూట్ చేసుకొని మాట్లాడండి సార్, అన్మ్యూట్ చేసుకోండి. మళ్ళీ మ్యూట్ లో ఉన్నారు. సార్ అన్మ్యూట్ చేసుకొని చెప్పండి సార్.
Secondary,
ఇప్పుడు చెప్పండి సార్.
sir, each single tRNA shows a extensive internal base pairing and acquires a clover leaf-like structure. This is also clover leaf-like structure of the DNA, RNAs. It is also stabilized by hydrogen bonding between the bases and is a consistent features of the this leaf-like, also leaf-like structure, clover leaf-like structure of the primary RNA structure. The secondary structure, the clover leaf structure, is all tRNA contains 5 main arms of the loops. tRNA 5 arms, 1, 2, 3, 4. Also 5 arms are there. So then acceptor arm, anticodon arm, DHU, also got a dihydrouracil. Then the TQS means thymine, pseudo aramide cytosine, also extra arms, which is also secondary strains of the RNAs are there. But this is also secondary. The secondary structure of the tRNA, then also called a CCA tails are there. Yeah, this one. This is also got tail of yellow acceptor stem. This one, yellow color acceptor stem. In purple variable loop. In orange D arm, orange color D arm. Red anticodon arm. In blue with anticodon. In black T arm. In green. This is also got a structure of the then tRNA is also differentiation of the mRNA and also tRNA differentiations are here. But this is also the tertiary. Then first one, the RNA primary, secondary, tertiary. 3 types are there. The primary, which the primary RNA and secondary RNA, tertiary RNA, which can be classified based on the structure of and nucleotide chaining. The tertiary structure of a tRNA, the L-shaped tertiary part is there. Then L-shaped tertiary structure is formed by further folding of the clover leaf due to the hydrogen bonds between the T and D arms. T and D arms of the, but the base paired double helical stems jet arranged in the 2 double helical columns, continuous and perpendicular to the another. That is also tRNA parts. But the types of the RNA. So this one, the structure is also the easily structure, primary, secondary, and also called tertiary. The 3 parts are there in structure. Then the based on the structure, the RNA which can be classified, they say majorly 3 types: mRNA, rRNA, tRNA. Additionally, 4 types are there. So these additionally 4 types which can be called this one. Then additionally other, the types of RNA in all prokaryotic and eukaryotic organism, 3 main classes of RNA molecules exist: messenger RNA, mRNA. That is first one. Then second one, transfer RNA, also called a small d RNA. Then ribosomal RNA means rRNA. These are 3 types major. The other are small nuclear RNA, snRNA, other type. Then microRNA means miRNA. miRNA, small interfering RNA means siRNA. siRNA, heterogeneous nuclear RNA means hnRNA. This is also 4 plus 3, 7 type of the RNAs which can be classified based on the structures. Then so this is also, first you go that ribosomal RNA, means that is also got a rRNA, third part. Third part is also, first you take the ribosomal RNA. This ribosomal nucleotide acid is the RNA component of the ribosome and is essential to protein synthesis. Only rRNA means ribosome means protein synthesis. The function of the ribosomal RNA molecules are necessary for ribosomal assembly and seem to play a key role in the binding of mRNA to ribosomal and its translation process. The ribosome is a majorly translation. It can measure important for the structure and development of the RNA structure. In the recent studies suggest that an rRNA component performs the peptide transferase activates thus, and it is also got a ribosome enzymes. So these enzymes are major important role in protein synthesis. That is called a character features of the rRNA. It consists of the predominantly material within the ribosome, which is approximately 60% of RNA and 40% of proteins by weight. The total RNA, ribosomal RNA means ribosomes are also got 60%, proteins are 40, the total 100 weight of the ribosomes, which are rRNA. Ribosome contains two major rRNAs and 50 or more proteins are there. Then the ribosomal RNA is from a two subunits, the large subunit LSU and a small subunit SSU. The LSU or rRNA acts as a ribozyme, the catalyzed peptide bonds formation. This is also when major, then also called a ribosomal after messenger RNA, mRNA. This mRNA is a comprised only 5% RNA in the cell. Then rRNA is more percentage in the RNA, but messenger RNA only 5%. Most of heterogeneous in size base sequence, all membranes of the class functions as messengers. Messengers means due to the information, the cell information is also carries a one cell to other cells. That is called a messenger RNA. Then it can information of the gene to the protein synthesis machinery. Then protein synthesis also machinery takes place. Then structure of the mRNA, the 5-end terminal end and capped by the 7 end. It is also methyl guanosine, and a triphosphate is a cap. The two, and the RNA is a parts, and the nucleotides are also called a packing to each other. The cap is involved in the recognition of mRNA by the translating machinery. It stabilizes the mRNA by protecting it from the 5 exonuclease enzymes. So this is also then structure character of the mRNA also completed, but this one structural characteristic of mRNA, which is also cap AUG, adenine, uracil, guanine, uracil, adenine, guanine, polynucleotide. It is also eukaryotic mRNA molecules, but it can be coding sequence, that is A to G, that is starting and stop the code. Then 5-end cap and 3-end tail impact stability to mRNA by protecting from the specific exonuclease, parts. Then it is a mRNA is also a major character features. Then it can perform in the machinery role in the transcription parts. Then next part also got a rRNA completed mRNA, but now tRNA. The tRNA is transfer RNA are smallest of 3 major species of RNA molecules. They have 74 to 95 nucleotides or residues, smallest percentage. But first one, ribosome percentage or more. Then the ribosome observe that tRNA, transfer RNA is less. But here is also got a messenger RNA. Messenger RNA is also less. This is also, they are synthesized by the nuclear processing of a precursor molecules. They transfer the amino acids from the cytoplasm to the protein, synthesizing machinery. Hence, the name is also tRNA. The transfer of amino acids, the major function of this one is also transfer. That is also called a tRNA base, transfer character base to the denoted the tRNA. They are easily soluble, hence called the soluble RNA or sRNA. They are also called the adapter molecules, hence they act as the adapter for the translation of the sequence of nucleotide of the mRNA in the specific amino acids. There are at least 20 species of tRNA, one corresponding to each of 20 amino acids required in the protein synthesis. This is also important of the transfer RNA. Then the primary RNAs are also divides the three types between tRNA, mRNA, rRNA. But secondary structure of the RNA, it can accepted arm. The accepted arm is also 3 and to the base of the 7 in. The end sequence is a unpaired cytosine. The cytosine adapted 3 end. So this is the tRNA bound with the amino acids are called amino active enzymes. So this is also then the secondary structure of the RNAs are base. Then the secondary structure of RNA is anticodons part, but here parts are there. tRNA anticodons. Here codon, anticodon, intracellular base pairing. This is also tRNA structure. Secondary structure of the tRNA, but secondary structure of it can base important of the DHU arm, or also this is also secondary arm. It is also complete of last. Then tertiary RNA structure. The tertiary RNA, the L-shaped tertiary structure formed by the holding of the clover leaf due to the hydrogen bonds between T and D arms. The base pairs are double helical stems get arranged in the two double helical structures. So this is the continuous and a perpendicular to the another cells. But that is also ribosomal RNA also completed, but then these are all is also over. Then here is also heterogeneous nuclear RNA. First then the primary, secondary, tertiary. Then in the primary or secondary, tertiary is also which can and a conversion of the one RNA to other RNA. But in the types rRNA, tRNA, mRNA, sir. This is a base. But here heterogeneous nuclear RNA, hnRNA is another type of the RNA. Additional RNAs in mammals nuclei, hnRNA is the immediate product of the gene transcription. The nuclear product is heterogeneous in size, variable, and is a very large. Molecular weight may be more than 10 to the power 10, while the molecular weight of the RNA is less than 2 into the 10 to the power 6. Then 75% of the hnRNA is degraded in the nucleus. So this is also HRNAs are differentiations. Then HRNA means heterogeneous nuclear RNA figures. Then it mature RNA formed from the primary transcription by the capping of this what? Capping. Then, but this is also then small RNA. Then hnRNA outer small RNA. Most of these molecules are complexed with protein and from ribonucleoproteins and are distributed in the nucleus, in the cytoplasm or the both. They range in size from 20 to 300 nucleotides and are present in 1 lakh to 10,000 copies per the cell. This is also small type of the nucleus of RNA. Then second one, third, first one hnRNA, small RNA, then small nuclear RNAs, snRNAs. So snRNAs is a subset of the small RNAs are significantly involved in mRNA processing and gene regulation. Of the several snRNAs are U1, U2, U4, U5, and U6 are the involved in a intron removal and a process. This is also U7 also is involved in the production of the correct process. This is also one character. The small nuclear RNA is snRNA. Then snRNA is involved in the process of splicing process. Splicing. So then microRNAs. MicroRNAs also got mRNA and small integrated RNAs are sRNAs. It is also additional parts of the RNAs. These two classes of the RNAs represent the subset of small RNAs. Both play important role in gene regulation. It is also MIR, RNA, siRNAs cause inhibition of the gene expression by decreasing a specific protein production. That is also characters of then microRNA. This is also called a microRNA. Then small integrated RNA, but here is also small integrated RNA. Then this is also significance of the mRNAs. But now differentiation between the RNA and DNA. So then what are the RNA and what are the DNA? What are the differentiation characters of these two? Then the RNA can easily destroyed by alkalis to cyclic diesters of mononucleotides. Then DNA resistance alkyl interactions are there. Then RNA is a relatively unsoluble molecules, undergoes easy and spontaneous degradation. DNA is a stable molecule. The spontaneous degradation is a very too slow. The genetic information can be stored for years together without any change. The mainly cytoplasmic, but also present in the nucleus. But here is a mainly found in the nucleus. Extranuclear DNAs are present. The base contains the varies from 100 to 5,000. The size is a variably. The DNA is a millions of base pairs are there. Depends on upon the upgrading. This is also then ninth one. Then there are various types of RNAs. mRNA, RNA, tRNA, snRNA, siRNA, miRNA, hnRNA. These RNAs are perform different in specific functions. But DNA is always a one type and performs the function of storage and transfer of the genetic information. The genetic information is a storage and transfer of genetic information from one generation to other generation, only two characters. But here RNAs are several types, but it can perform the specific functions. The no variable physiological form of the RNA are found. The different types of RNA do not change their forms, but the form is also stable, does not change of RNA. There are a variable form of DNA, A, B, and Z. It can be changed, not it can modify of DNA. But RNA is a synthesized from DNA. It cannot be DNA except by the action of the reverse transcription. It cannot duplicate the except in the certain virus where it is a genomic materials. DNA can form a DNA by replication. It can also form a RNA by transcription. So many copies of RNAs are present per cell, but single copy of DNA is present for cell. This is also major differentiation of the RNA and DNA process. Then this is also then amazing DNA facts. So what is amazing means the DNA from a single human cell extended a single thread for almost 2 meters long. But very, very amazing process. Each single cell thread of human being DNA is 2 meters. It contains a information equal to some 6 lakhs of printed pages of 500 words each. In the library, about 1,000 books. You know that some of the 1 lakh, 6 lakhs of printed pages of 500 words each. That is got an amazing feature of the DNA that is important of the. So it's revised that process, but it is also got a filling the evidence. It is a major important. The RNA stands for ribonucleic acids. You know that RNA is usually single strand. The sugar present in RNA is a ribose. The nitrogenous base uracil in RNA replaces the thymine. Thymine is replaced to the uracil. It is also a major point. There are 3 main types of RNAs: mRNA, tRNA. That is also got 3 types. 3 types are there. But here, the mRNA carries the genetic code from DNA to the ribosomes. That is got mRNA answer. Then dash brings amino acids to the ribosome during photosynthesis, means tRNA answer. That dash is a major structure and a function components of dash ribosomes. In answer, RNA and ribosomes. rRNA is also major. Then transcription is the process of making dash from DNA templates, RNA forms, transcription. Then RNA plays a key role in the process of dash, majorly protein synthesis. That is also base or introduction to the RNA, look around 10 days, feeling the structure of RNA. So then is also 10. RNA is made up of a long chain of dash nucleotides. It is important. Each RNA nucleotide contains a dash and a nitrogenous base, means ribosugar phosphatase group nitrogenous base. Types. The nitrogenous bases found in RNA are dash, means adenine, guanine, cytosine, and uracil. Thymine replaces the uracil. That is important. RNA contains the base dash instead of dash, means uracil. Uracil is instead of thymine in RNA, which is found in a DNA answer. Then the sugar in RNA is ribose, which has one more oxygen atom, then deoxyribose in DNA. Answer means ribose and oxygen. But RNA is usually dash single stranded, while DNA is double stranded. You know that simple single stranded RNA and double stranded RNAs both are based on the sugar molecule structures. Then the backbone of RNA is formed by alternating sugar and phosphate groups. Then in RNA, dash are pairs with dash, means adenine, thymine, or adenine, thymine means in a DNA, but here RNA is uracil, that is pair, adenine, uracil, guanine, cytosine, which is a pairs for the RNA templates. Through the hydrogen bonds during base pairs, the three-dimensional structure of tRNA resembles as dash shape, clover leaf shape. It is important. Clover leaf shape of structure of RNA, that is called dimensional structure. RNA molecules can fold into the complex structure, that is called intramolecular base pairing, which is also important of the then introduction or structure base of RNA, which can be displayed in the 10 type of the filling the blanks. Next types of RNA. Then it is also got a 10. The three main types of RNA, you know that mRNA, tRNA, and rRNA. mRNA stands as messenger RNA. tRNA transport dash amino acid to the ribosome. That is important. Then tRNA stands for, rRNA stands for ribosomal. mRNA is carries genetic information from DNA to the ribosome. It is also one character of the, then tRNA has an anticodon region that pairs with the codon on the mRNA. mRNA answer is the anticodon. Then rRNA provides the structure and enzymatic support for protein synthesis. Then snRNA means small nuclear RNA. It is a small nuclear RNA is involved in a mRNA splicing. That is called a snRNA, mRNA splicing, three type of the dashes. miRNA means microRNA. It helps regulate gene expression. Answer, miRNA means siRNA dash is used in gene silencing. Is important. Means siRNA is important of the gene silencing through RNA. Then that is important of the types of RNA. Then last point, functions of the RNA. Then these functions are which the primary function of mRNA is to carry genetic information from DNA to the ribosome. That mRNA in ribosome, genetic information. Then dash helps in translating the mRNA code by bringing the correct dash, means tRNA. The tRNA helps translating the mRNA code by bringing the correct amino acids during protein synthesis. Then dash provides the site for protein. tRNA is a site for protein synthesis. It is also one function of the RNA. Then RNA is involved a dash, means RNA gene expression by controlling which genes are turned on or off. Then dash and dash are types of RNA that regulates a post-transcriptional gene cells. Post-transcriptional gene means miRNA and siRNA means post-transcriptional gene translating process. RNA can also function as an dash enzymes, and such RNA molecules are called ribozymes. Ribozymes are special type of the enzymes which can be performing the RNA molecules, and it means RNA can be functioning as enzymes, means ribodage or ribozymes. snRNA is involved in a splicing of a pre-mRNA to form a mature mRNA. That is, RNA dash is the enzyme that synthesizes RNA from a dash. Means RNA polymerase is the enzyme that synthesizes RNA from a DNA templates. Answer RNA. In viruses like retroviruses, dash can serve as genetic materials. RNA as genetic materials in viruses, that is called retroviruses, instead of DNA. That is also important of the filling the blanks of the function of the RNA, which is all is also important for the character features of the RNAs. But here, this is a which are a clover leaf model. This is also type of the color, the primary and secondary tertiary, but here primary structure, secondary structure, and tertiary structure. In secondary is also called a clover leaf structure, is a clover leaf. All tRNA contains a, this is also acceptor arm anticodon form. This one, acceptor anticodon, A means anticodon. Means also called a TP, and in the thymidine part, this is also thymidine part, this one. Here A, here D. D is also called a dihydrouracil. Dihydrouracil, this part. Then extra arm, this is also extra arm. This is the important word. Then it is also one type of the mRNA molecules or codes, tRNA codes. This is also tertiary RNA. Primary, secondary, and tertiary, but here there is a primary, secondary, but here is also this tertiary structure. Tertiary structure of RNA. This tertiary structure, the L-shaped tertiary structure is formed by the further of the clover leaf shape structures. Clover leaf shape. The base of the pair double helical structures. But this is then the types of RNA. But here is the other RNAs are important. 1, 2, 3, 4. The 4 RNAs are important. Small nuclear SN. Small S. Nuclear means N. SN RNAs. Then micro means MI RNA. M afterward I. Small interference means S and I. SI. First one S. N. Here is also SN. SNRNA. But here SR or SI. Small interference. Heterogeneous nuclear means here HN. This is also basic parts of the type of the RNAs. Actually, 3 is also major. Messenger RNA, transfer RNA, ribosomal RNA. Only 3 base. But in additionally, which can be classified the 4 types. That is important of the character features of the RNAs. RNAs. This is a this is also all of the completed the RNA. In the last class, DNA over now RNA is also completed. This is also fill in the blanks. First your revision of this. This is also more amazing part. Amazing part that can be great for. Here is filling the blanks of RNA interactions. Interactions only RNA interaction that in major fill in the blanks are it can be given here. But this is also structure of RNA. Structure of RNA is also 10. But here is types of RNA is also 10. But here the filling the blank functions of RNA. The total, then the total 40 filling the blanks are given topic, which is also important for the studies and easily prepare in the PG interns, also jail interns. Here is also one basic structure. It's also differentiations. The differentiation of single standard RNA, but here double standard DNA. The single standard DNA, double thymine and uracil, both are differentiation of thymine. This is also thymine, but here is a uracil. Thymine, uracil, which is also U and also T. It is a various. Then DNA to RNA differentiation is a majorly single standard of RNA, and also thymine is instead of the uracil. The cytosine, guanine, adenine, uracil. Here is a cytosine, guanine, adenine, thymine. That is also called a nucleobases of DNA. But here is a nucleobases of RNA. RNA versus of DNA. But here ribose nucleic acids. Here is also deoxyribonucleic acids. Ribose and deoxynucleic acids. But this is to the complete of the topic of just. This is also small nuclear RNA. This is an RNA. Is lastly, we are complete the